All right, everyone. Thank you so much for joining us. This is yet another journal club hosted by the Cannabis Chemistry Subdivision at the American Chemical Society. My name is Andrew Pham, and I'll be hosting this very special holiday edition of Journal Club. Usually we are on the last Thursday of every month, but since November and December are both holidays, we're going to do it today. And today we have a very special guest, Emily Kirkham. I've known her for years, and I'm so glad we could be reunited in this format today. Emily is the founder and president at National Laboratory Services, a Canadian company focusing on analytical laboratory consulting for the cannabis industry. She specializes in method validation, quality control, and training for licensed producers and cannabis testing laboratories. She's recently partnered with Rudra Testing Solutions to further diversify her consulting services. She has a bachelor's in chemistry from McGill University and has managed method development and analysis in environmental and government laboratories. In 2014, she founded her own development business and brings over 22 years of experience to the NLS team. Today, she's going to be talking about forced degradation studies and how it can provide a shortcut to avoiding quality issues in cannabis. So Emily, go ahead and take the reins.
Thank you, Andrew. Thank you so much for inviting me here to present to the ACS Cannabis Subdivision. I've enjoyed very much your other presentations. I feel like I'm in excellent company with these presenters. And your introduction was great. A little background about me, as Andrew said. I started in the cannabis industry back in 2014, supporting companies in Canada, North America in quality assurance and quality control. And recently partnered with Rudra Solutions and CARO Laboratories, which is based on Vancouver Island. And this data presented in this presentation was made in collaboration with these companies. So the title of this presentation may get some people's knickers in a knot, because when you say shortcuts, that really maybe bothers some of the scientists out there. But the business and money people in the industry are probably salivating at that idea. The key here is that we are saying shortcut to avoiding quality issues in cannabis. And that is the key. What we want to try to do is to help people avoid running into quality issues unprepared and having them smack you in the face with expensive recalls. See, the money people would already be happy again. And this is really, the cannabis industry is kind of like that, right? How we merge having a successful business while keeping efficacy, safety, and quality at the forefront. So that's what we're going to discuss today. So now this slide may seem like a no-brainer. Of course, there are risks when you have quality products, problems with your products, but it's really important to highlight what can actually happen in the real world, all across the spectrum. If it's a low risk, you could have product loss, time loss. On the medium, recalls and brand damage. And at the high end, even illness and death. So here, I'm just going to highlight a couple of examples that you may be familiar with. This is the E-Valley crisis that happened back in 2019, where vape products were contaminated with what was suspected to be vitamin E acetate that led to 40 deaths and over 2,000 people with severe illness, as reported by the CDC. And another case here of Delta-8 THC products leading to adverse reactions and even one reported death of a child. Here, consumers were likely over-consuming Delta-9 THC. And there are many more types of these examples. Unfortunately, without companies fully understanding their products, there's going to be more of this to come. So what we're going to talk about today is like how we can avoid these. So how do we fully understand our products? What this presentation is going to focus on is product degradation, understanding how your product behaves under certain circumstances. Packaging, manufacturing, storage, excipients, and how all of those can affect the product quality. So when I say drug substance, this is the product that you're creating. And I know that sounds like a very medical standpoint, but really I would argue that the majority of cannabis could be used for medical purposes, even if the user did not get a prescription per se. It may be for stress and relaxation or mental health or any number of reasons. So the quality of adult use or recreational products is just as vital as for medical cannabis. And in my opinion, it should be subjected to the same investigations and quality requirements. And as you will see in this presentation, there's issues such as reduced potency, change in color, odor, taste, shelf life, and even the formation of toxic impurities. So we talked about degradation of the products. Now we're going to get into forced degradation and what that is. So this is an essential tool in product research and development, and it's often, in our experience, it's often not done and it's overlooked and not used. So what are forced degradation studies? So basically you put products, your drug substances or your API through a stress test. Generally, they would be more stressful than what you would normally be subjected to. But you can take that information and interpolate that into what will happen in a real world situation. We do that in an elevated stress so that we can see these results faster. It's all about the kinetics of the reactions. And we'll go into detail on each of these stress factors and how to evaluate them and why they are important. So discovering these issues early can lead to your commercial decisions. How do you package a product? Do you ditch a product altogether? Maybe you need to look at the additives. All these questions, when you know the answers early on in the process, it saves you countless hours and money. So do you actually need forced degradation data? Well, in some markets, yes, it is absolutely required if you want to sell products in many international markets, as it's part of the ICH quality guidelines. But also there are tangible and intangible issues that you can avoid. As I mentioned before, identifying issues early saves you money in the long run. It's also important in brand building, because if you have a shelf-stable product, your customers are going to be happy and they're going to come back for more. So now we're going to get into the nitty-gritty of what this actually looks like. So in forced degradation, there are 4 main stressors that are used. We have acid-base, oxidation, photolytic, and thermal stress. So I'm just going to talk a little bit about these 4 individually. So here in this case, we're looking at exposing your product to acid-base stress. So let's take an example of a CBD beverage, which is common, and we see this. You want to make this beverage taste like Coca-Cola. So perhaps you add citric acid, making the product slightly acidic. You package this product, you get it on the shelves, everybody's super excited about it. People are consuming it, they love it. But 6 months later, 6 months it's been on the shelves and the customers are complaining and saying it doesn't taste like Coke anymore. Now it tastes like Mountain Dew, or worse, Pepsi. And we've seen this happen before. It might be the color or the odor, where the product suddenly turns purple and smells like fish. No one will ever buy that again. I'm sure you've had the experience of buying a product that tasted off. Did you ever buy that product again? I know I wouldn't. So what happened? Doing an acid stress test beforehand will tell you the answers to these questions and help you avoid them. So we'll get into that, these specific examples a little bit later on. So here we have oxidation. In the oxidation studies, we often expose the product to hydrogen peroxide and evaluate the product for potency and impurities. So again, for example, say you have a THC or CBD product and you're making it with MCT oil. It could actually be the MCT oil that undergoes the oxidation, creating impurities in the product. Sometimes these stressors are linked. So this process could be linked to an acid-base reaction because the impurities created from the oxidative stress on the MCT oil may actually have double bonds, pi orbitals, in which case they can donate electrons, creating a Lewis acid or base. So you may end up with the same issues, the same degradants that came from the acid-base reaction, but it's actually a result of oxidative stress on just the MCT oil itself. So how do you avoid this beforehand? So if you run the stress test, you'd have to actually run it on your CBD and THC product and on the MCT oil separately and understand those reactions independent of one another. So the third type, we have photolytic stress, and there's actually a specific ICH guideline for the photolytic stress, whereas the rest of these are all sort of under the general ICH quality guidelines. In this case, you have a photolytic chamber. It's like a box, like an incubator with white fluorescent light and near UV light, and you monitor the exposure with a calibrator. The guidance tells you how many photons of light you need to have the product subjected to. And this procedure evaluates your product against light exposure. And certainly, I'm sure many of you are aware, cannabis is no exception. It's not photostable. So photooxidation occurs in most cases. And this is the primer for what kind of intermediate or secondary packaging you would use. For example, like how do you protect your product from light? Even in processing, you may be, mixing your THC API into a vat of, your excipients, your oil or whatever you're making, and then the room has lights on and it's open and the whole area is exposed to light. This stress test would give you an idea of whether or not this is a bad idea to have that process, and maybe you need to limit that light exposure. So finally, we have thermal stress. Thermal stress actually gives you the best indicator as what's going to happen to your product while it's sitting on the shelf. In this case, the product is exposed to elevated temperatures and then evaluated. For example, you would run your product at least, you'd expose it at 3 elevated temperatures. And using kinetics, you can predict what will happen at room temperature. So this uses the Arrhenius equation, and I'm not going to get into the math here, or I might put everybody to sleep. But basically, for every 10-degree increase, you double the amount of interactions that occur in the reaction. So you can plot this. From the slope, you can ascertain the activation energy for the reaction, and allowing you to back calculate to room temperature or 25 degrees, because you know the rate of the equation. In one example from this test, we predicted that a new impurity was formed from a THC product. And this impurity would actually co-elute on your chromatogram with CBD. So over time, it appeared as if THC was decreasing and CBD was increasing, when in fact, it wasn't CBD. Now, this is what's defined as a non-stability indicating method. Having a stability indicating method that that when a stress happens, that you're correctly identifying the CBD or the API and the impurities, that they're resolved and quantitatable. So you wouldn't want to be testing a product that sat on the shelf and you think that it's actually like developing CBD when it's not. So now we're going to get into the reasons why we want to perform these forced degradation studies. So here we want to characterize the drug substance. We're talking about the behavior of the molecule when it is stressed by these different factors. What is the mechanism of the degradation? And if you know that, you can avoid it. Maybe one mechanism has a different rate, or maybe one has different impurities. The mechanism is the pathway of the degradation, and it's important that we evaluate this. Quite often we think we really understand the stability of plant products. I've been talking a lot about oils and vape products and other products like that, but in the plant it's important as well. We might have one cultivar like a pink kush and a white widow, and they have maybe similar cannabinoid profile, but the rest of the phyto environment is different. So the terpenes and the flavonoids. So you actually need to evaluate the degradation of these two products separately, because there's also reactions with the rest of the phytoenvironment that can be significant. So here, we want to determine the structure of the degradants. And here we show 3 common degradants from cannabinoids that you may be aware of. They may be generated by acid base or thermal stress. And I'm sure you're familiar with the psychoactive compounds degrading to non-psychoactive CBN and delta H THC. And this is a result that, obviously consumers don't want. Most consumers are looking to get high. We need to avoid this degradation because they're going to be getting a less potent product as a result. And in the case of the compound that's shown here on the right, CBD is degrading to a toxic compound, hydroxyquinone, under basic conditions. So we would have found this if you had done an acid-base stress test. So this is a well-known pathway. It's actually an anti-tumor drug. So in one case, people are not getting high, and in the other, they're actually consuming a toxic compound. Neither of these do we want, obviously. So another thing we want to understand is the degradation pathways. So knowing this, you expose THC to oxidative stress, and you will form CBN and delta 8 THC. Obviously, again, this is a well-known pathway, but clearly we do not want products to degrade to less potent components. And again, some more examples that we talked about previously of oxidation of neutral phytocannabinoids and their respective quinone species, and conversion of CBD to CBD quinone from a base catalyzed oxidation. And here, under acidic conditions, CBD forms delta 9 THC. Again, not something we want. If we want to claim that a product is CBD only, we want to ensure that the product is actually stable in that form. For medical purposes, this is clearly evident. We don't want a child with epilepsy receiving a drug product that has the wrong levels of delta 9 THC or CBD. So how do you avoid these stressors? Maybe it is an event or packaging material or process that is forming the impurities that you're seeing. These experiments can also help you avoid overdoing something. For example, you may find out that oxidative stress is not really a concern. So then why would you waste all that money nitrogen packaging your product? It's important to understand the impurities that are going to be formed in any manufacturing process and how you can control the levels of those impurities if you're going forward. So impurities in any product are important as they could be toxic and potentially lethal. Knowing them is the first step to controlling your process. So we are going to have a few case studies that we have examples that we've seen in the real world that we're going to show you. So here you have a API like THC or CBD. You get a C of A, it says 99% pure, and you have a few impurities that you know about. You have specifications on that, like in CBD, you'd have your CBD-V is always there and CBD-C4. Or in some case, maybe you had, like this could be also a THC oil product, it was packaged, you put it on the shelf, the method was validated for the testing of this product. 6 months later, you're getting complaints that the product is not effective anymore, and you're getting adverse reaction reports. When you go back and analyze that product, you see that there's an unknown peak and that the active is actually degraded. So suddenly the method that you validated is not valid anymore, and you have this unknown impurity. So if you had done the forced degradation studies, you would see that pure impurity beforehand, and perhaps it's a degradation of photolytic stress. So you go back and get better packaging, and you prevent exposure during your processing. This happens quite a lot where you analyze your products a bit later on and you get an impurity and you don't know what's going on, but you could have explained that if you had done the testing beforehand. So we're going to go back again to, the reasons why we should perform these forced degradation studies. So we need to determine the intrinsic stability of drug substances in formulation. And what this means, I'll give you an example. Let's say you made a lotion and the main component is THC, but lotions have a bunch of excipients. Sometimes there's like 15 of them. So you need to stress test on the base formulation without the active THC. And then say you have that some excipient made an impurity and it co-elutes with your THC. And what we've seen is that when you calculate the THC, when you first make the product, say it's 10 milligrams per dose. And then later on, when you analyze it, it comes back at 50 milligrams. And that's actually over the limit for your regulatory requirements. I know there's lots of different jurisdictions out there, but this is an example we've seen before. And the product is thrown out, but it's actually not that the THC is higher, it's that you didn't have a stability indicating method to know what that degradation product was, and it was actually co-eluting with your THC, and your THC was actually stable. So you just threw a bunch of product away that you really didn't need to. So again, this is similar to the example we just said before. We want to generate stable formulations. So the forced degradation studies, if you don't do them beforehand, that allows for a quick dismissal of incompatible ingredients. You want to do this before your product is on the shelf. And in the case of medical cannabis, before you launch into expensive and time-consuming stability studies. So another reason is that we want to optimize our products and formulations. Maybe there is something you can add to it, like an antioxidant that will help with the product stability. So if you can know that beforehand, there are mitigating factors that you can put in place to prevent these issues from happening. So again, I mentioned this before, where you had a method validated for your product and that worked. And then 6 months later, you find out that the method isn't valid anymore. And all that time and work is wasted. So this could be the specificity of your analytical method. And it's about having a stability indicating method. You need to do your stress tests on the API and on your placebos or your formulation, because you need to know if the impurities are coming from your API or from your formulation so that you can properly quantitate them. And we're going to go into another case study here, which goes towards this having a proper stability indicating method. So in this case, we had a potency result, and it was acceptable. And over time, we know that THC is not stable and it will degrade. It is as expected as a North American soccer team not making it into the FIFA finals. Sorry, but that one cuts deep for us Canadians too this year. But what you find when you analyze your product later is that the THC is not actually decreasing. And the marketing people are all jumping for joy. They're like, oh my God, the product is so stable. But the chemists are suspicious, and we should be, because we know that the THC should degrade. That's just what happens. So what's going on here? In this example, this was from the excipient degrading, which created a product that co-eluted under the THC peak. So you might not actually see the THC decreasing. It's actually, having this co-elution problem. So, you have customers coming back and saying, this product isn't getting me high. I expect it to be, a certain level of THC, and it's not. It's not actually what's shown on the label. And if you had this problem, you're trying to sell a product in Europe, it was tested over there, and they could see that the THC was actually decreasing and it was going below their, the acceptance criteria, it would fail. It wouldn't be approved. We've also seen this when we talk about that In the first slide, I talked about the Delta 8 THC vape products that actually had the Delta 9, or they were, edible products and other such things. They actually probably had Delta 9 THC in them, and the method did not resolve those two peaks. And that was the issue with that. It's potential anyway. So if you had conducted the forced degradation study on this formulation beforehand, you would have known that you would have tested your excipient separately. You would have seen that it had a degradation peak, and then made sure that your method could resolve those two peaks, so you wouldn't have had this problem. So we're going to go back to why we need to perform these degradation studies. So we were talking about this. We want to establish a stability indicating method, and this is really critical here. A stability indicating method must accurately measure the changes in the THC or the CBD concentration without interference from other degradation products. And as you can imagine, if you didn't know these peaks were all co-eluting, then how would you be able to accurately determine the concentration of your active compound? As a chemist, if I validated a method and later, like 6 months later, they come back to me and I found out that it was actually looking at 5 overlapping peaks, I would be very embarrassed and have to go back with my tail between my legs and redo a ton of work. So here we're talking about, again, the stability indicating method. We need to ensure that our peaks of interest are completely resolved. Sometimes you may need 2 or 3 completely different analytical methods in order to properly quantitate your product. The forced degradation studies will show that to you before you put your product on the shelf. So finally, also we can use these forced degradation to predict the stability of your product. And we talked about that when we looked at the thermal stress. That's quite often a predictor of shelf life and stability. It helps you understand what's going to happen in the future. Over time, things will degrade. Performing these stress tests will give you a window of what's going to happen in 24 months. So you won't be surprised. How fast has your THC degraded? What are the kinetics of that? So a little bit here, we're going to talk about formal stability studies. So if you're making a medical product, this is what's required for determining your shelf life. And it can be a 2 to 3 year process. Forced degradation can give you the data in less than 1 month. You can't actually use it in most cases for getting a product into clinical, but you can get that information, and it's going to tell you, is this going to work, before you waste 3 years of time and money. Just for one product, as an example, it can cost up to $100 grand just for the testing for formal stability. And that doesn't include all the time and resources. It can be up to like $1 million. And that would all need to be thrown away if 2 years in you discover that, hey, your product isn't stable and it's actually making a toxic impurity. Now you have to start over and go back to the drawing board, reformulate your product, add mitigating factors, prevent UV light or thermal stress or oxidative stress. All this stuff is important to know beforehand. So just a little slide here about what's involved in a formal stability study and why that takes so much time. Your product has to be tested for, potency, micro impurities, up to 36 months. According to ICH, you have to have this data to calculate your shelf life, as we mentioned. And there can be multiple categories, like you have to do room temperature, refrigerated, accelerated. If you start this formal study without understanding your products from forced degradation, you could be wasting a ton of money. So again, this is just reiterating how important it is to know what will happen to your product under stress. It will help with development, scale up, optimization, packaging, and storage. You just need to know this stuff before we put products out on the shelves. And here, this is a summary slide. So we've been talking about this through most of the presentation, but here we are showing two routes that you can take. Process one, here, you have the marketing team. They have this brilliant product, this wonderful, exciting THC beverage. The marketing people rush it to production and get it onto the shelves, hoping to sell it quickly in time for Christmas, because who doesn't want to have a THC drink when all your family is coming over for the holidays, right? But you put that product on the shelves in August, so it gets out there and it's time to get it into the shelves. By December, that drink tastes like garbage and no one will buy it. So you've wasted a whole bunch of time and you've gotten a product out there and you don't even know why it went bad. In the second process, the marketing team gets the same idea. That product goes to R&D. They look at the forced degradation studies. They discovered that it needed an antioxidant, or it just needed to be protected from photolytic stress. After that, it went to scale up. Instead, they packaged it in amber bottles, or they put nitrogen blanking on it. And the product was wonderful. And it sold amazingly well, and the company made a ton of money, and everybody was happy. So happy ending to this story. The business people and the scientists are happy. And that's what we're looking for here. A way to predict and prevent quality issues before they occur. So that's it. Thank you very much for your time. And if you have any questions, I'm happy to answer them.
Thank you so much, Emily. Yeah, everyone, as I mentioned in the chat, we have about 15 minutes or so to do Q&A. If you have any, feel free to type them into the chat or into the Q&A section. But I guess I could go first. I was wondering if you had, without naming any names, any really great quality horror stories. I just want to feel some schadenfreude, as a analytical chemist. Just being right for once, saying, I told you so, money people. Sorry, I think that's just something I really want to tap into.
Really great horror stories. Well, I can say that like the worst horror story really is that you do all that work of putting products through formal stability, and I have experienced that, and only to have to go all the way back to the beginning, and the scientists were telling them, well, this isn't going to work, this isn't going to be stable, it's going to degrade, and then it starts to degrade and they have to throw it away, like a lot of it away. And nobody likes to throw cannabis products away because they're super valuable and all the time and effort that went into it. So I know it's not very specific, but there's not really anything that I can say.
Well, you don't have to be
without
specific.
Giving something away.
But yeah, that definitely happens a lot. And I just glad to hear that we are worth our salaries by preventing this type of loss.
Absolutely.
So thank you, scientists, for holding your posts. Any questions in the chat again? Let's see what else we got. Well, it didn't seem like anyone has any questions. So in that case, if you guys have any other questions, you can email Emily directly or reach out through the American Chemical Society's Cannabis Chemistry Subdivision. Again, if you're looking for some sort of analytical consulting, Emily Kirkham is one of the best in business. Her email address is right here, emily@nationallaboratoryservices.com. And yeah, thank you so much for sharing everything you did today, Emily.
Thank you so much, Andrew, for having me. I really appreciate it. Looking forward to future presentations I can watch with you guys.
Of course. As a reminder, this webinar is recorded and we will be uploading it to the Cannabis Chemistry Subdivision's YouTube page if you ever want to review it. And I want to wish everyone happy holidays and have a great new year. See you next year.